Okay, so I'm going to post a quiz for you by 7 tonight. Don't email me about it before 7. And you are going to have from that time until about 1 o'clock, slightly before 1 o'clock the next Monday to do the quiz questions. All open notes, open book. There's no reason you shouldn't get 100. If you, however, forget to do it, I'm not going to negotiate with you. That'll be a life lesson, right? You're like, oh, it's past 1 o'clock. Because 1 o'clock is when the class before you guys starts. So I want to be able to discuss all of those answers. After you get it done, FYI, after you submit it, you can't go back and change your answers. You can change them as many times as you want until the quiz is done. And you don't have to do them all at the same time. You can go look up 2 of them, do a couple answers. You can say, I don't know this one yet, do the next one. Does that make sense? Most of them will be over material we already covered. There'll be a couple that's over the last part of this set of notes. You can still do it, right? If it's over the last part. And then on Monday, we'll finish up this set of notes and start the next one. So this is the last set of notes that really overlaps with 1610 very much. But we're going to do it slightly differently. Okay, so I know that you've talked about channels and carriers and active and passive transport and osmosis and endo and exocytosis and all of that business in 1610, right? But my objective for you today is to be able to predict when you would use one versus the other. Instead of, for instance, just telling me that's a channel, get to the point that you can predict when you would use a channel, a carrier, when you'd need energy, when you wouldn't, right? So that's the objective. And then, of course, we're going to apply that to situations that exist in the human body, and it's going to be like with us all semester long. So we will run into a transport mechanism in the kidney, and I will say, this is using a channel. Why wouldn't it use simple diffusion? Why is it not osmosis? Why is it not this, right? Okay, so we're going to start by talking about your friend, the plasma membrane, which you've talked about in excruciating detail in Biology 1610. So before we go any further, you guys remember what the plasma membrane is mostly made of, so tell me, what's it mostly made of? Exactly. And it is a boundary between the intracellular fluid and the extracellular fluid. But it's not an impermeable boundary, it's a selectively permeable boundary, and you've heard that a million times. And selectively permeable is like a really arbitrary bouncer in a nightclub, right? So I'm a bouncer. You guys want to get into the club. Whether you get in or not depends on what's already inside the club, what my needs are, right? Go to a fancy Manhattan nightclub, and unless you're incredibly hot and got a whole bunch of money, you probably ain't getting in, right? If you are smelly and belligerent and also male, you ain't getting in at all, right? And under what circumstances would you get kicked out? Well, depending on whether there's too many people inside the club. So the criteria are not unchanging for the nightclub any more than they are for the cell, right? There are some things that can probably almost always get in. Very hot women and very scantily clad hot women, I should say. Glucose, for instance. No, I'm just kidding. Oxygen, really. So we are going to talk about the differential or selectively permeable nature of the membrane. So let's start trying to predict. So when you're looking at what gets in or out of the cell, what do you have to consider? The first thing is size. What do you think? If you were trying to get something into the cell, here's the ECF, here's the ICF. Size matters, and in this situation, if you're trying to get something into the cell, is it more likely to go in if it's large or it's small molecule? Very small things, small, right? Teeny tiny things have a much easier time getting in, like a little oxygen molecule, O2, versus a whole E. coli cell, right? It's going to have a much easier time getting in, so size will matter. And then the next thing you will need to consider is the electrical charge or polarity of the solute that you're trying to get in. So, of course, the phospholipid bilayer is lipid. And so lipid soluble stuff does whatever the heck it wants related to the cell, in and out, right? What about charged things, things that are charged or polar? Do they tend to be water soluble or lipid soluble? They're water soluble, they're not lipid soluble. So what would happen if our friend sodium has a high concentration outside and a low concentration inside? So when given the opportunity, it would go into the cell, except that it can't. Yes? Because, of course, it's charged, right? So it can't go in, can't go straight through the cell membrane. Does that mean we'll never get sodium in? No, it means we're going to have something else to transport sodium in. That makes sense? So the way that you typically get really small charged things in, like ions, is by providing an ion channel for them, okay? And the channel makes this little bitty space that's just big enough for something to go through the cell membrane, and it's not lipid right there. For instance, if this is a sodium channel, it might even have a slight negative charge on the inside, so it's more attractive to sodium. Yeah? By the way, channels can't really be specific to one ion, but they can prefer one, right? So it's a sodium with an asterisk channel. It also lets other positive ions through in a pinch, yeah? But you can still call it a sodium channel. It just doesn't exclusively transport sodium. So electrically charged, or polar ions or molecules can't go straight through the lipid portion, but they can go through, quite often, channel proteins. Channels are only good for tiny things. They are not good for big molecules. So if you start moving up larger molecules, this is, for instance, a channel protein. Usually ions are about as big as they can go. Not always, but usually. Okay, so that's a channel protein. Now, the next thing we have to consider after size and charge is other types of solubility. So, of course, we have lipid-soluble stuff and we have water-soluble stuff. Lipid-soluble stuff goes wherever it wants in the entire body. So if I had oxygen, which is a nonpolar lipid-soluble molecule out there, and it was low in here, it goes in when it wants, and you can't stop it, right? Which is actually good in some ways, because do we have to worry about oxygen moving inside of the cell if we get it close to the cell? No, it's just going to move straight in, because oxygen is usually pretty low on the inside of the cell, because it gets used up by what process?
Cellular respiration.
Cellular respiration, exactly. So if you get oxygen anywhere near the cell, it's usually going to go straight in. Good, because it's lipid soluble. What other kinds of things are lipid soluble? Alcohol is lipid soluble. That's the reason if you drink a couple sips of wine on an empty stomach, how long does it take before you feel it? Just like a couple, 30 seconds, just long enough to get there, right? It doesn't have to ask permission to go into your brain, and if it did, I would say, okay. So go straight in. And then fatty acids, steroid hormones, and anesthetics, guys, topical anesthetics, those wouldn't work if they were not lipid soluble. Does that make sense to you? Okay, all right. So water-soluble substances. So when we get to water-soluble substances, after we've already considered charge, let's consider a couple other things. If it's a teeny tiny water-soluble substance like water, it might be able to move in and out without asking permission. Although, FYI, you guys know that water moves in and out of cells based on its concentration gradient. But we used to think that it's just kind of sneaking through. And then they figured out, I don't know, 10, 15 years ago, after I had this stuff, they figured out that there are actually a bunch of these little tiny channels called aquaporins. Right? Aquaporins? Yeah. But there's so many of them that basically it looks like water's just moving out in and out willy-nilly. Right? So water moves across. It looks like it's just moving straight through. What about carbon dioxide? Because carbon dioxide is also a small water-soluble substance. It moves in and out of the cell willy-nilly too. I kind of wonder whether given another 10 or 15 years, we might not find a whole bunch of carbon dioxide channels. But for right now, it looks like it just can sort of break the rules of solubility. It's small enough, okay? So if it's a pretty small water-soluble substance like water or carbon dioxide, it looks like it's moving straight in and out down its concentration gradient. What if it's a mid-sized water-soluble molecule? What am I talking about, mid-sized? Well, H2O is relatively small, easy to count those atoms. C6H12O6 is mid-sized, right? Or any of the amino acids are mid-sized, right? They're not so big that I can't count them at all, like a protein that's 300 amino acids folded together. That's a large water-soluble molecule. But a mid-sized one, like glucose or a single amino acid, they can't go straight through, right? Because they're not lipid soluble. They're too big for a carrier. A lot of times they are bigger than the carrier, okay? So what's plan C? If they can't go straight through and they can't use a carrier, plan C is, oh, sorry, I said carrier. They can't use a channel. If they can't go straight through and they can't use a channel, plan C is to use a carrier. So I want you guys to listen this time because I don't want you to think that you can use the terms channel and carrier interchangeably. They are different terms. So what is a carrier? What a carrier is I have C6H12O6 out here and it wants to get in, and I want it in there, and the concentration is low of glucose inside because I've used it up by cellular respiration, right? So it's pretty dependably going to be low inside most cells. So I want it in there. Can't get in this way. Can't get in this way. It can get in by a carrier. And what a carrier has is, let's make glucose this shape. Let's find a carrier that has this binding site for it. And then the carrier is going to dump it inside the cell. But it's going to do it by actually holding onto it and moving it. So this is how the carrier works. Carrier has a shape-specific binding site for the thing that's going to move, right? It is going to bind to that thing. The binding of the substrate to the carrier will actually change the shape of the carrier such that you will dump that inside. The dumping changes the shape of the carrier again such that you reposition it for the next one. Does that make sense? Okay, so this is a carrier protein, yes, has a shape-specific binding site, okay? Now let's just think about this for a second. Now we have three different transport mechanisms. We have this one, which is simple diffusion, right? We have this one, which is just diffusion through a channel. And then we have this one, which is diffusion through a carrier, which is called facilitated diffusion. And let's just number them. So let's say this one is number 1, this one is number 2, this one's number 3. Which one's fastest? Number 1. And why? There's no limitation to the places that it could occur, right? And which one's next fastest? Channel, right? Because it doesn't have to bind, change position, bind, change position, bind, change. This one's the slowest, yeah? In addition to being slow, let's see if you guys can conceptualize this. This one, because it has a shape-specific binding site, is going to be subject to saturation. So let's talk about this for a second. This is simple diffusion, okay? With or without a channel. So as the concentration goes up, the net flux goes up. So basically, if I have a greater concentration gradient, then I have a greater transport rate. What I'm saying here is that if I have 10 molecules of oxygen versus 1, then I have a relatively slow transport rate. But if I have 10,000 versus 1, I have a relatively fast transport rate. That make sense? No? It doesn't? Okay, if I have 2 and 1, how quickly is it going to move? Not very fast. Now I have 200 and 1. It's going to move really quickly. Got it? Okay. And that's true whether you use a channel or not. A channel will just take a little longer to get there, right? Okay, now I want to look at what would happen if you had the same story, only you're moving through a carrier. So stick with me, okay? Again, I'm going to have concentration gradient, and I'm going to have net flux, which is how fast you move, the transport rate, okay? And as the concentration gradient goes up, the transport rate is going to go up until what occurs? Saturation. What is occurring during saturation? The carrier is full, yeah. So it doesn't matter if I added more glucose, right? I still couldn't get it to move any more quickly because the carriers are subject to saturation because they're shape specific, right? They get full, yeah? It does simple diffusion through the membrane have to deal with saturation? No, it doesn't have to deal with saturation. Does that make sense? Okay. What's the only way? You know the answer. The only way that you could increase the net flux or transport rate, not enzymes, increase the carriers. But same concept, right? Okay. So you will see really, really commonly that anything with a shape-specific binding site is subject to saturation. Anything. Okay. All right. So, all right. And that will matter to us in a little bit. I'll tell you a couple of ways in the human body that it matters. Okay, so That's carrier. So what about the last thing? What if it's too big to do any of these things? So I have an enormous protein, right? It's 350 amino acids. It's some kind of crazy folded structure. You know it can't do simple diffusion, right? And it can't go through a channel because it's probably bigger than a channel, and it's generally bigger than a carrier. So what is plan D? Plan D to get sent, is endo or exocytosis, depending on which direction you were trying to send it, right? And endo and exocytosis are super slow and they're very energetically expensive. So I want you to imagine that I've got this enormous protein, or maybe a bacterium. So I'm a coccus bacteria because I'm round, and this is my cell membrane. And I need to get it in, right? Need to get it into a white blood cell so that I can lyse it and whoop up on it. So what I'm going to have to do, too big to go any of the other ways, so I'm going to have to fold my cell membrane around it, right? And then pull it inside the cell, right? Slower than Christmas and super duper expensive, okay? So why does it matter? Because the prediction mechanism that you're going to use all semester long to figure out what transport mechanism you would use is the cell is going to do the cheapest, fastest thing it can, right? And if you are not using simple diffusion, you better be able to defend to me why you're not. Right? If you were trying to put something through a carrier instead of a channel, you better know why, right? If you were using endo or exocytosis instead of any of those other cheap ones, you better know why. So what we're going to do is, as we go through the semester, we're going to be like, okay, this one is going to do exocytosis of the kidney, and why couldn't it use any of the others? And you're like, oh, it's enormous. You can't get rid of it by any of those other mechanisms. Or why would you need to use energy? We're going to start talking about energy in certain situations. So cheapest, fastest way first is what you're going to use as your prediction mechanism. And we're going to start to use this right now. All right, so let's start talking about simple diffusion. We can do simple diffusion relatively quickly because you've had it a million times. So what simple diffusion is, guys, is simply movement of molecules, solutes, pretty much anything, from areas of their high concentration to areas of their low concentration. And if you go over time, start out like this and just let simple diffusion occur, what is this going to look like over time? What will this look like after some time? It's going to be straight across, right? Okay. What's happening? Do those little molecules have some directionality and intelligence to know which direction to go? No, they're just smashing into each other until they all have equal space, right? They're all equally distributed. Just Brownian motion, right? It is passive transport. It doesn't require ATP, right? And it will move things from their high concentration to their low concentration, although none of the molecules has a net direction that you could depend upon. You couldn't, for instance, say, George right here is definitely going to end up on this side. Right? He could stay on either side, right? And will it work through a membrane? If the membrane is permeable to the solute, it will work through the membrane. If the membrane is not permeable to the solute, then it won't, okay? So the net movement is from high to low. Distance matters. Diffusion is really pretty effective over microscopic distances, meaning that oxygen is all right here and the cell is right here. It's going to get inside, right? However, the oxygen in the alveoli of my lungs are not going to get to my little toe by diffusion alone, right? It's just not an effective transport mechanism for long distances. And then it may or may not involve a membrane. And you don't need to put energy into it. So let's think about a couple of situations in which you've experienced diffusion. So let's go back to making Kool-Aid. Ain't nobody make Kool-Aid anymore because you've noticed that you have to dump a cup of sugar into the pitcher. But that was no problem when we were kids. Did you guys drink Kool-Aid when you were a kid? Oh my, and nobody makes Kool-Aid. So we're going to drink some fruit punch Kool-Aid because it's the best kind, right? And we're going to make it, only we're going to be super lazy. And what we're going to do is we are going to put the Kool-Aid packet and a cup of sugar. And we are not going to stir it, right? So we don't want to burn any calories before we eat that cup of sugar. So we're going to just leave it on the counter. Question for you. If you leave it on the counter for long enough, will it become Kool-Aid without you stirring it? It totally will. It's just diffusion, right? If you just leave it there for long enough, the entire pitcher will be red Kool-Aid, right? And it will be nice and sweet. And you don't have to do a damn thing to it because it's just moving from high concentration to low concentration. as long as you got time enough to wait for it. The same thing you can think about the last time you took a family road trip, especially if you have children. I have an 11-year-old boy. I generally have like anywhere between 2 and 6 11-year-old boys in my car at any time. And you know that they fart. You know that they do, because that's what they do. And when they fart, does it all stay right here? It certainly does not. It moves from high concentration all the way up to the low concentration in the relative piece of the front, right? And it will move even if I put a mesh barrier between me and them, right? Because the mesh barrier is permeable to the solute. And does my little boy have to do this to make it move? No, no energy is required, although he does do that to make it move. He definitely does. I don't know when farts stop being funny. I don't really know. Apparently not 11. They don't? Somebody needs to tell everybody that about having boys, because there's a lot of farting that goes on. Okay, so, if you are moving something across the cell membrane, what is the effect of increasing the amount of cell membrane? So let's just give 2 situations, A and B cell. I've got something that is moving across the cell membrane. Which one's going to have the faster transport rate? If it's moving across the cell membrane? Yeah, B, right? You've got more cell membrane. It's going to increase the transport rate. So microvilli will increase the transport rate, which is why you have microvilli cells with microvilli in your intestine, for instance. Okay, last little tidbit is that sometimes you're not just dependent on concentration gradient. Sometimes you're also dependent on charge. So let's talk about this. Okay, so when you are moving an ion, concentration gradient matters, but also electrical gradient can matter. Because if I'm trying to move sodium from its high concentration to its low concentration, yeah. If it's moving toward a neutral, it goes however fast it goes, right? What if it's moving from high concentration to low concentration toward a negative? Is that going to increase or decrease the speed of transport? Increase the speed of transport, right? Because positives are attracted to negatives. Now, if I am moving a positive sodium down its concentration gradient toward a negative, what's going to happen to the speed of transport? You're going to slow it down. Does that make sense? So there are 2 things that have to be considered when you're moving ions. There is the concentration gradient and the electric gradient. They call those collectively the electrochemical gradient. Okay, the electrochemical gradient. So how do you figure out which direction it's going to move? If it's got an electrical gradient that's favoring one direction and a chemical gradient that's favoring the other direction, well, there is an equation that you can do called the Nernst equation that will teach it to you. And we may get to the Nernst equation later this semester because there's one situation in which you can use it. Well, there's a bunch of situations. But there's also a way to fudge it that will work almost all semester. And the way to fudge it is a little bit of a fib, but it's a useful fib. And that is, for the vast majority of your physiology time, what will be the best predictor is that the concentration gradient will determine the direction and the charge will impact the speed. Okay? Now, there is a situation in which that can flip, and when that occurs, I will tell you. Okay? And when it becomes necessary to do the Nernst equation, we can do the Nernst equation, which puts it all in together and helps you figure out. Okay, but for most of our time, the concentration gradient will determine the direction and the charge will impact the speed. Okay? And put an asterisk there because it's only truthy or truish. There's like exceptions to that. Okay, so now let's talk about channels for a second. So if you don't do simple diffusion and you diffuse through a channel, it's just called diffusion through a channel. And there are a couple of different kinds of channels. There is a channel that is always open, and a channel that is always open kind of looks like this, and it's called a leak channel. And it's called a leak channel because when given the opportunity, things will leak through them, right? They don't have the capacity to close, okay? You will run into boatloads of leak channels in cells, and at first it's really hard to figure out why the hell they exist, but later on you will see why they exist, but you can't close them. So a leak channel is always open. And then we run into another type of channel, and the next type of channel is called a gated channel, And a gated channel, I'm going to show you what a gated channel looks like. And what I really want you to think about is how is this gated channel different than a carrier, okay? So a gated channel, so let's draw a cell. So here's my cell. Here's my, let's make this a sodium lead channel. And sodium will move in and out down its electrochemical gradient. Whenever given the opportunity, it will move. Okay, you can't ever close that one. That's just a lead channel. And then I'm going to draw a gated channel. I'm going to draw two different states of the gated channel. So here's the default state of the gated channel. This is a gated sodium channel. Okay, this is the default state. Sodium's not going anywhere, and neither is anything else. So its default state is closed, okay? But you can open it. Once you open it, it behaves like a leak channel, okay? So this is the next state of the same channel. So, and then sodium can go in and out. So how do you open it? There are several ways to open a gated channel. The first type of gated channel that we will see has a shape-specific binding site for a chemical messenger. But the chemical messenger does not go in. It just binds to and opens. This might be a hormone or a neurotransmitter or a paracrine agent that says transport sodium. So now I'm going to ask you, in your words, what is the difference between this, which is, by the way, called a ligand-gated channel or a chemical-gated channel? What is the difference between that channel and a carrier? Go ahead.
The carrier is allowing the thing in that it matches with, and this is allowing something else in.
Exactly. So that's the difference, okay? The only way that this would be a carrier is if it actually bound to sodium and let sodium in, right? It binds something else which will allow it to let sodium in. Does that make sense? Okay. So this means that although we want sodium to move in freely, we don't want it to move in freely all the time. So we can say not now, right? Does that make sense? Okay, so this is called a chemical or ligand-gated channel, and the chemical or ligand that you use to open them are very frequently chemical messengers. Like what? Name some categories of chemical messengers.
Like the sodium, potassium.
No, the chemical messengers that would bind.
Hormones.
Hormones. Insulin is one of them, yes. Other categories of chemical messengers besides hormones. Neurotransmitters, paracrine, and autocrine. So one of the things that chemical messengers often message about is, hey, increase your ion transport. Okay, I'm going to open an ion channel for you. Does that make sense? Okay, so that is a chemical gated channel. There are other kinds of gated channels that are important. One of them we will see all semester long. I have no idea how to draw it. You've seen the limits of my drawing capacity. So this one is called a voltage gated channel. And a voltage gated channel, you have the cell sitting there minding its business and the state of its channels are closed. And then you zap it and the change in voltage opens the channel, right? And then you get the ion movement. Now you think, why the hell would you go zap into anything? Well, you zap yourself all the time because you have body systems that are based on electricity. Which body systems? Nervous and all of your muscular systems. So don't be at all surprised that we find voltage-gated channels in nervous and muscular tissues. Does that make sense? Okay. And then the last type of channel we don't really run into very often, but it's kind of important for athleticism and coordination and proprioception and just knowing your position of your body in space. And these are called mechanical gated channels. And they are channels that open when you squish them or pull them on the cell. So picture this. You fell asleep, you wake up, it's pitch dark, you can't see a thing, and your arm is right here. Here's my question. How do you know your arm is right there? Because this tendon and ligament are pulled. They are pulled, right? And that sends a certain signal by opening channels, right? and these are squished, and that sends a different signal. And the relative pulling and squishing of different ligaments and tendons and the channels within them tells you the position of your body in space. It's called proprioception, right? And it has a lot to do with these mechanical gated channels. And some of you are incredibly coordinated. And you have a really, really good proprioception. And how does that occur? Well, a combination of the luck of the draw, right? You might just have the genetics and anatomy that's good for proprioception. But then you also reinforce that by, for instance, I don't care if you're the best kickball player when you're 7. It doesn't mean that you're actually going to be a professional soccer player unless, of course, you train that proprioception. And really what gets better, by the way, is not the channels here, but the interpretation in your brain, right? Primarily in your cerebellum. It gets really, really good at interpreting the signals of proprioception really, really quickly. Mine is terrible. I'm incredibly clumsy, right? And I'm the only person in my family who can't play music, which is also proprioception as well, like the feedback of playing music, physically playing music. Okay, so now, so we've done osmosis, we've done the types of channels, and now we've done diffusion, we've done the types of channels, and now we're going to do osmosis. But I know you've done osmosis before, but for some reason, I do not know why. People memorize osmosis, but they do not understand it most of the time. So what we're going for today is we're going for understanding. And the reason we're going for understanding is because memorization will not help you when you have to predict which direction water will move in the kidney, because you actually have to understand the nature of osmosis. So I'm going to teach it to you, and hopefully I'm going to teach it to you with a slightly different spin than you've heard before so that it will stick, or maybe it already stuck, and you will just confirm that you are brilliant. So let's talk about osmosis. Osmosis is just a special instance of diffusion. It is diffusion of water and water alone across a selectively permeable membrane, and the selection criteria are solid. Here's the selection criteria. Bouncer, yeah? Water is a hot girl. She's always getting in and out when she wants to, right? The solute in this situation is the belligerent stinky dude. And under no circumstances is he going to cross the membrane. Does that make sense? So water will always be able to move across the membrane, and the solute will not be able to move across the membrane. Since it's diffusion, guys, does it require energy? No, it does not require energy. So it's passive transport. It does require a selectively permeable membrane. It does not require energy. So it depends upon a concentration gradient, but here is the weird thing. It depends upon a concentration gradient, but the real concentration gradient that's important is water's concentration gradient, and you are never given water's concentration gradient. So here's the issue. What you will have is a situation in which you have a non-penetrating solute, okay, also sometimes called impermeable, that can't move, okay? It's too big, there's no transport mechanism, it's water soluble, you can't get it, for whatever reason, it's not gonna get across. And then you will have water. If you ever see this, or this, you should have a bing in your head that says osmosis, okay? Because you can't do diffusion. So let's look at a couple of examples, then we'll talk about human examples. So let's look in the book, okay? In your book, here's a figure, okay? So what we're gonna try to do is predict in these situations where water is going to move. Okay, so here's the deal. These little purple balls are solute, and they are non-penetrating solute, and I have to tell you that or else you won't know what to do. Because if the little purple balls were soluble, wouldn't they just diffuse out? Yes? Okay. So I'm telling you it's a non-penetrating solute, which makes you know that you can't move the solute. Okay. The problem is that they always give you the concentration of the solute. They never give you the concentration of the water, but the solute doesn't move. Okay. So here's the deal. You have to get used to sort of flipping around the data that you're given. So this has 300, let's say, milliosmoles, but it doesn't really matter. Kumquats, whatever, it doesn't matter. As long as you're using the same inside and outside, you can compare big and small. So it's got 300 milliosmoles of solute, 150 milliosmoles out here, okay? So you're given the solute concentration, but what's really important to you is the water concentration, because wherever the solute is, the water is not. Yes? Okay. So in this situation, what I need you to know is that water is going to move down water's concentration gradient. So in this situation, where is water higher? Outside the cell. And water is going to move down water's concentration gradient inside the cell. Does that make sense? Okay. The solute didn't go anywhere. Okay. The relative concentration of the solute might have changed, but the concentration of the solute didn't change. It's exactly the same number of molecules inside as there was before. Is that okay? So, all right. So now let's look at this situation. Okay. Whole bunch of belligerent stinky dudes out. Only a couple inside, right? He's not moving, okay? So in this situation, where is the water concentration higher? Inside the cell, yes? Because wherever there is solute, there is not water, yes? So it's higher on the inside of the cell and it's lower on the outside of the cell. So water moves down water's concentration gradient, yes? So water is going to move outside the cell. Does that make sense? So here's what people generally do with osmosis. They memorize. They say, oh, in an isotonic solution, it doesn't move anywhere. In a hypertonic solution, it does this. But that won't help you when you run into two very different solutions and you just have to look at the water. So we need to get to the point that we understand it. So what we're going to do is we're going to practice it a little bit. Okay, so if we have a selectively permeable membrane, water which can cross the membrane, and a solute which can't, we're going to do what you've probably done before. We're going to be good scientists, and we're going to set up an experiment in which we vary a single variable, because you don't vary more than one at a time. So I'm going to set up 3 beakers, A, B, and C. And in each one, I'm going to put a cell, make it big enough so that you can see. The cells are all identical to one another, and cells aren't empty. So I'm going to tell you that X is the non-penetrating solute. So cell, same cell in each one. And then I'm going to put the same volume of solution in A, B, and C. And I'm going to vary the solute concentration in A, B, and C. In solution A, I am going to put 0 non-penetrating solute. I'm just going to put it in straight water. In solution B, I'm going to try to balance the solute concentration outside with the solute concentration inside. Okay? And so I know it's not representing concentration, but just go with me. And then in C, I'm going to put it in a really, really concentrated solute solution. Okay? Okay. You ready? So I'm going to ask you the same questions for all 3 solutions, and then we will name them. I want you to remember the rules. The rules are that water is the only thing that can move, and water moves down water's concentration gradient, right? So in situation A, the question is, which direction is water going to move? Into the cell. Now somebody raise their hand and tell me in your words, why? Why is water moving inside the cell? Go ahead.
The cell has less water inside of it than it does outside of it.
Yes, exactly. Less water inside than outside. Okay, so what is the impact on the size of the cell when given a little time? Yeah, it'll get big. If it gets big enough and it's an animal cell, and yours are, it can lyse, cause lysis. In a plant cell, it doesn't do that. You can pour straight water on a plant cell all day long, and it will get bigger and bigger and bigger, but they have a cell wall. So what happens? Your plant needs water, you water it, stands up. Doesn't cause lysis. Good? Okay. And B, same 2 questions. Which direction is water going to move in B?
Nowhere.
Can't move nowhere, because I told you that the hot girl goes anywhere she damn well pleases. No net movement. Yes, exactly. It does go in and out, but no net movement. What's the impact on the size of the cell? Yeah, even if I didn't draw it the same, it's the same size. Okay. And then last but not least, in this situation, which direction is water going to move? Outside the cell. Yeah? And somebody else raise your hand and tell me why. Why does water move out of the cell? You guys are so quiet.
Because the concentration has to be equal, so all of the little guys are going to move into it, which is going to cause the cell to shrivel up.
The guys are going to move into it?
The little, you said they're stinky guys.
No, but the solutes don't get to come in. It's just that the water leaves. Yeah. Yeah, the solutes are non-penetrating. So somebody else give it a shot. Go ahead.
The water's going to leak outside of the cell in order to even out the water concentration.
Yeah, it moves down water's concentration gradient, right? So water is higher relatively on the inside of the cell than it is on the outside of the cell. So water moves down water's concentration gradient. So now, last question is, what's the impact on the size of this cell? Yeah, what's that called? Crenation. Yeah. So, and then let's name the solutions and talk about some human examples of the solutions. First off, here is a crenated red blood cell, which does not travel through capillaries well. Okay. Now, what we need to do is we're going to name the solutions, and then we're going to talk about human examples of each one. Okay. So when you name the solution, you name them blank tonic, and tonic has to do with water pressure, tonicity. And the prefixes you use are iso, hyper, and hypo. Okay. And here's what you're naming. You're naming the solution because that's what varies, right? Isn't the extracellular fluid far more likely to vary than the intracellular fluid? Okay. So you're naming the solution with reference to the cell. Does it have more solutes, the same amount of solutes, or less solutes than the cell? That make sense? Okay. So the prefix iso means the same. So which of these three solutions has the same amount of solutes inside and outside? B. Yeah. The prefix hyper means above or more than. Which of these solutions has more solutes than the cell? C. So hyper. And the prefix hypo means below or less than, and A has less solutes than the cell. Good? Okay, how I remember this from 7th grade when I first learned it is, you're hyper, do you get skinny or fat? Skinny. Hypo, hippo, big fat cell. And then the other one, I don't know. Didn't need that. I didn't need 3. I only needed 2. Okay, so let's give some human examples, and then I'll show you guys a video of this occurring. So, in an isotonic solution, I would bet you that you have isotonic solutions in the vast majority of places in your body, because do you really want your cells to be swelling or shrinking? So I would hazard a guess that unless something is really wrong with you, your plasma is isotonic to your blood cells. Yes? Almost every extracellular fluid that you're going to run into is isotonic to the cells that it is around. Good? And also, let's think about this. I'm super duper 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 dehydrated, speaking of that, and I go to the hospital and they give me an IV. They do not give me an IV of straight water, do they? They give me an IV of normal saline, which is blank tonic to my blood. Mm-hmm. Does that make sense? Okay. Now, a hypertonic solution, when you give a hypertonic solution, it's going to cause crenation. It's going to cause crenation. So let's talk about that. You're stranded on a desert island. You can't find any fresh water. Why do you not drink the salt water? It will dehydrate you. Let's talk about the mechanisms that cause that dehydration. I drink the salt water, and as I drink it, it comes into contact with all of the epithelial tissue in my digestive tract. And it comes in contact with all of those cells. What does it do to each of those cells? It pulls water out of each of those cells. And then I get diarrhea, right? And it comes straight out the other end, and I end up having less fluid in my body than I had before I drank it. Does that make sense? So it's a hypertonic solution. In that situation, you're actually completely dehydrating yourself. So you don't want to drink salt water. You should drink your own urine before you drink salt water, but make sure it's yours. right? Because you already like got all those little beasties, probably, right? Don't drink anybody else's urine. I mean, it's just a good general policy. That's why you took physiology. Oh, I don't drink other people's urine. And then a hypotonic solution. A hypotonic solution is when the solute concentration outside is greater than the solute concentration inside. With red blood cells, if you, for instance, shot yourself up with straight water and you could keep yourself from peeing, you could actually cause hemolysis and kill yourself that way. I mean, it'd be kind of a dumb way to do it. There are probably really easier ways to do it, but it's totally happened. When you sit in the tub, by the way, for long periods of time, what you do is you are sitting in a hypotonic solution, which is kind of counterintuitive because when you get out, you think that your fingers have shrunk, but they have not. They are swollen. And it takes a little while because you've got a layer of keratin, so you're kind of waterproof or you're really water resistant. But what happens is the water eventually starts to move in because you're sitting in a hypotonic solution. But do you remember that your skin is spot welded to the basement membrane by hemidesmosomes? Do you remember that? They're little spot welds, but they're not everywhere. So what happens is you swell, but you don't swell everywhere. So if you got a really good scale, you actually weigh slightly more when you have soaked in the tub than before you got in. So that'll get you some trivia points sometime. Okay, so let's look at a little video that shows you how that occurs. Where is that video? Got a video that shows you all of this occurring. There it is. Okay.
Differentially permeable membranes. As a result, they tend to be isotonic to surrounding fluids. In other words, they have the same salinity. Blood cells in normal saline solution have a slight dimple. When those same cells are exposed to a less saline solution, the cells begin to absorb water, going plump. If they absorb too much water, the cells will burst. When those cells are then exposed to a more saline solution, the cells lose water, becoming wrinkled and shriveled.
So that's crenation. So you at least get to see it happen. When we used to be able to deal with like real human blood, they were super limited about letting you guys deal even with your own fluids. We're still trying to get permission for you guys to do that in phys lab. But you could actually just see that. You could put your own blood on a slide and just squirt some salt water in it and watch them shrivel up and then squirt some straight water in it. I'm still trying to get permission, but they do not want to give it because they think you're going to give each other diseases. Well, just don't touch anybody else's stuff. Just like hold on to your pee, right? When you're glucose testing it. Hold on to your blood when you're doing it. It's fine. But they say no. Although every other school does it. All of the other colleges use real human blood and real human urine in classes. Okay, so that's osmosis, and now we are going to move to mediated transport systems. What do I mean when I say mediated transport systems? FYI, this term is used differently by different textbooks and by different people, so it's just important that you know what I mean by it. A mediated transport system is when I'm trying to get something from point A to point B, but I use a carrier to help me. So not a channel. I don't mean channels when I say mediated transport. So the general information is that mediated transport will use a carrier protein, back to our little friend, the carrier protein that we saw back here. So all the way back here. So we're going to use a carrier protein to move something from one side of the membrane to the other, okay? And since it's a carrier protein, you're going to run into a couple of things. First off, remember carrier proteins have saturation limits. And then second, I didn't tell you guys this before, but it should be relatively intuitive. If you have a shape-specific binding site, you are subject to regulation by other molecules. Like for instance, in this situation right here, let's say this is a glucose carrier, right? You with me? And glucose wants to go inside the cell, but the default state of this glucose carrier is that it does not fit glucose until something binds over here, insulin. Yeah? Insulin activates or unlocks the glucose carrier, right? So then it works. So does it surprise you that since enzymes are shape specific and can be regulated by regulatory molecules, carriers are too. So you can change what the carrier is doing by turning it on and off. Does that make sense? Okay. So it's subject to regulation by other molecules. Okay, so a couple of types of transport that are mediated transport systems, and the first one is facilitated diffusion. Facilitated diffusion is simply diffusion through a carrier. And so let me tell you all you would need to know about this picture to know it was facilitated diffusion. If I told you that glucose was high here and low here, then this would be facilitated diffusion, right? Because it's going from high to low through a carrier. Yes? Okay. Is facilitated diffusion faster or slower than simple diffusion? Slower. Is it faster or slower than diffusion through a channel? It's slower than diffusion through a channel. So why would you use it? Back to your critical thinking. Too big to use any of the other mechanisms. Does that make sense? So when do I use facilitated diffusion? You use facilitated diffusion very frequently to get nutrient molecules for catabolism or anabolism inside your cell. So how do you get an amino acid inside the cell? Usually by facilitated diffusion. How do I get glucose in? Usually by facilitated diffusion, right? They're mid-sized water-soluble molecules. I need them for stuff. I move them in by facilitated diffusion. Is that okay? And I can change the transport rate by binding something to it and turning it on, turning it off. Okay? So that's facilitated diffusion. All right. Doesn't require ATP, moves things only from high concentration to low concentration. So it's just diffusion through a channel. And then the last of, well, no, not the last, almost the last. The transport mechanism that we're going to do next is the one that you really, 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 really need to understand because it's going to be important for the rest of the semester. And that is primary active transport. So in this situation, what we are going to do is we are going to try to get something for the first time that we've talked about today to move against its concentration gradient, right? And if you want to move something downhill, you don't have to pump it and you don't need to put any energy into it. But if you want to get something to move uphill, you need to pump it. So you're going to need to get energy into it. So if you want to move something against its concentration gradient, you're going to require energy. So primary active transport is when you move anything. It doesn't matter if it's an ion or small molecule or a relatively large one. To get it to move against its concentration gradient, you are going to need a pump. What is a pump? A pump is a carrier, so it has shape-specific binding site, plus ATP. That's what a pump is. Carrier plus ATP. So the net effect is that you are going to get an accumulation on one side of the membrane. So we said that if simple diffusion started out this way, and I just did diffusion, and you gave it just time, it would end up like that, right? Okay. What will happen with active transport? With active transport, if you start out like this, and it's concentration gradient against net flux or movement, okay? And then you did active transport. Depending on how quickly you transported it, you could either just maintain the concentration gradient, basically battling diffusion, right? That makes sense? Or what if you transported it more quickly? Couldn't you get an accumulation on one side of the membrane? Right? That makes sense? So with active transport, what you end up getting is a net accumulation on one side of the membrane. So if you want to maintain the accumulation, what do you think you're going to have to do? You have to keep pumping. For how long? Forever. Okay, that's what we're going to see in your cells, is that there are some pumps that are constantly active in every single cell that you have, because otherwise you will lose your concentration gradient for some ions. So the metaphor that I use for this one is, imagine that you've got a 24 balloon bouquet, you got a hatchback, yes? And you're going to try to get it into the hatchback. Only you're poor, so you're missing 2 of your windows. They ain't in there. Okay, so here's what we're going to have to do. We're trying to accumulate the balloons on the inside of the car. And so I'm pushing and pushing and pushing and pushing and pushing. And as long as I'm pushing, they're going to accumulate on the inside of the car. And as soon as I stop pushing, what's going to happen? They're going to go straight back out. But my windows are open. So now I got an extra problem. I'm pushing them in, they're going out that window. I'm going to pull it, grab it back in, going out that window, pull it, grab it back in. This is your cell. Constantly got crap leaking out, and you're trying to maintain a concentration gradient, right? And so if you want to keep your concentration gradient, you're going to have to keep your pumps going all the time. So I'm about to teach you the most important pump in your body. It exists on every single one of your cells, and you use anywhere between 40 and 60% of your ATP just running these pumps. And I know you've heard them before. They're called the sodium-potassium pumps, or sodium-potassium counter-transport pumps, or sodium-potassium ATPase pumps. It doesn't matter because they're all the same. So as long as it's got sodium and potassium, we're all talking about the same pump, okay? So here's the deal. The sodium-potassium pump is going to take care of a homeostatic need for the cell, of the cell. So we're going to look at it real quick. And instead of memorizing the sodium-potassium pump, we're going to build the cell and figure out why you need it. And hopefully you will understand the entire process. There's one little thing that I won't tell you yet, but you'll get it later. Okay, so in order to understand the sodium-potassium pump, I'm going to tell you just a couple of pieces of information that you're going to have to memorize if you haven't already done it, and that is this. So here's my cell, here's the ECF, of course, here's the ICF, okay? Now, how many of you guys already know, you don't have to, but do you already know where sodium is relatively higher, ICF or ECF? Yeah, the sodium has a relatively high concentration in the extracellular fluid relative to its concentration in the intracellular fluid. Good? Okay. And who knows where potassium is relatively higher? Yeah, relatively higher concentration in here. Oops, sorry, keep doing it. Potassium. And a relatively lower concentration of potassium here. Okay? Now, that will get you what you need. If you didn't already know that, you do need to memorize that. I'll give you specific numbers later, but the highs and lows you need to memorize. Okay. Here's something that's important. This is homeostatic for a cell. Okay. It doesn't mean you don't disturb it, but it means you always have to put it back, right? Just like saying your homeostatic blood pressure at rest is 120/80. It goes up, but you just have to put it back every time. Same thing with this. Okay. So we're going to have this as a goal. But then we are going to have a monkey wrench thrown in, and the monkey wrench is that this thing exists. There are windows that are always open in the cell. And they are, this is a sodium leak channel. They exist, they're relatively abundant. I know right now you're like, just close the damn thing. Later on, I'll be able to convince you that it's necessary that it stays open, but that's for next time, not for this time. Okay? So it is there. And when sodium is given the opportunity, which direction does sodium leak? Try again. In, yeah. Sodium leaks in, down its concentration gradient, right? Okay? And this is a potassium leak channel. Which direction does potassium leak when given the opportunity? Out. Okay, I know you know this, but verbalize it anyway. Why won't those leak in the absence of the channels?
Charged.
Because they're charged. Exactly. Good. So the channels have to be there for them to leak. So now, basically, we have a homeostatic goal, and then we've got something constantly jacking it up. Okay? So how are we going to maintain homeostasis despite this? What we are going to have is that sodium and potassium are constantly diffusing down their concentration gradients. And despite that, we manage somehow to maintain this approximate concentration gradient for sodium for every 15 you have outside, you have 1 inside. So that's your high to low. And for potassium, for every 30 you have inside, you have 1 outside. Now, I'm not saying we never vary from that, but we go in a range around it, and we put it back if we damage it. So how the hell do we accomplish this despite this? Well, we are constantly using this, and it's the sodium-potassium pump. So now, instead of me telling you, I'm going to ask you, if I want to maintain these concentration gradients despite the fact that this is happening, first question for you. Which direction do I need to pump sodium to maintain homeostasis? Sit there and think about it. Trying to maintain homeostasis. It's leaking in. I want it low. Pump it out. Yes. Pump it out. Good? Okay. Potassium. Which direction do I need to pump potassium? In. Yes. Okay. Okay. And to get things to go against their concentration gradients, and they are, right? I need to use ATP. When you use ATP, it becomes ADP. Got it? Okay. Now, to remember how much the rate at which it moves these things. Does anybody call it the potassium-sodium pump, or does everybody call it the sodium-potassium pump? Sodium-potassium. You guys are not talking enough today. So work on that for Wednesday. I'm flapping around up here, drawing pictures, and you get nothing. Sodium-potassium pump. If you call it sodium-potassium pump, then you can remember the ratios by calling it the 3-2-1 sodium-potassium pump. It moves 3 sodiums every time, 2 potassiums, and uses 1 ATP for the pump. So 3 sodiums, 2 potassiums, 1 ATP for the pump. Got it? Now, why? Why does it move those? Well, there's a couple of whys, but let's do the first why. The first why is simply because it's got binding sites for, look, here's your pump. 3 sodiums fit right there. Grab them, squirt them out. 2 potassiums fit right there. Grab them, squirt them in. 1 ATP fits right there. Does that make sense? So 3 binding sites for sodium, 2 binding sites for ATP, 1 binding site for the pump. Okay, so this is called primary active transport. And you have a figure, it didn't show up right here, in which you have a sodium channel, a potassium channel, and a pump in your notes. You should be able to predict in those notes which direction sodium is going to move through the channel, which direction potassium is going to move through the channel, and which direction both of them are going to move through that pump. Okay, so I want to show you an animation. Okay, ready? Okay, so this is our friend, the sodium-potassium pump. This video will both be on the video that's screen captured. It's also on the links page of melanieb.com. Whenever I've used videos in class that I have permission to share, they're on the links page so that you can like run through them and go backwards and forwards and listen to the narration and all that kind of stuff. All right, so this is the sodium-potassium pump, extracellular fluid, intracellular fluid. Sodium, potassium. You don't see ATP yet, but you will in just a second.
The sodium-potassium exchange pump.
I don't want to hear him talk. Okay, so I bet you can figure out what fits where. What fits there? What fits there? What fits there? Okay, so here we go. Sodium binds. I pop that phosphate off for the energy to get sodium to move outside. And please note that I simultaneously reveal the binding sites for potassium. And note that although I do not actually move sodium and potassium at the same time, their movement is coupled, right? Because if I moved sodium and didn't move potassium, can you see that that pump is useless forever, right? I actually have to move potassium after that, right? So how do I get the energy to move potassium? That phosphate that I was holding on to will fall off, yeah? And then I get squirt potassium back in this, inside. That make sense? So here we go, sodium out, potassium in, reset the pump. That makes sense? Okay, so that is the sodium-potassium pump. That pump is so freaking important that every single cell in your body has sodium-potassium pumps. You use between 40 and 60% of all of your cellular ATP just maintaining and running your pumps to maintain your concentration gradients. And you have body systems that screw up your sodium and potassium concentration gradients on purpose to send messages, and those just use the pumps more frequently. Okay? Did you have a question?
Yeah, do you only use the ATP when you're getting the sodium out? And then
Well, you actually use it in 2 separate stages. So the phosphorylation, the popping the phosphate off, moves the sodium out, but that phosphate is still high energy, and it falling off moves the potassium in. So you're basically not using all of the energy just in one fell swoop. Okay, so it's still got some energy in there as well. So the whole thing is energetically. It it uses 1 ATP per movement of the entire pump, right? Okay, and then you can rephosphorylate the ATP by the way too. You can
So
stick a phosphate.
getting that phosphate in there is what sends
The energy part really happens, by the way, right here, right there, phosphorylating that ATP off. Dephosphorylating that ATP, popping it off, is where the energy is. But you don't use all of that energy there. You've still got some of it for here.
And then the second half of it is when it's popping off, that's creating the energy for the...
Sort of. The real energy burst happened when you snapped it off. You just didn't utilize all of it in the first part. You still got some of it left, right? Okay. Everybody good with this? Okay. So this is your sodium potassium pump. Really important. Here's what I would think the good student would be able to do with the sodium potassium pump. The good student would be able to build this story with only these pieces of information. See if you think that if given some time you could do this. Could you build this every time instead of memorizing it, just starting with your sodium and potassium concentration gradients? Just think about that. If you just started with your sodium and potassium concentration gradients, couldn't you put in the channels and figure out which direction stuff would move through the channels? Could you do that? And then if you had the channels in, couldn't you figure out which direction you needed to pump? Does that make sense? Right? This is the kind of knowledge that is physiology knowledge, right? Instead of memorization, you actually have the level of understanding that you don't have to memorize it because you could figure it out every time. FYI, I've never memorized this. I build it every single time, right? And it never fails me because it is a story with a narrative that only makes sense if you get all the pieces right. Does that make sense to you guys? So that's the level of knowledge that means, hey, I'm doing really well. I don't really need to study. And then if I can ask you questions in which you understand terminology. So here are the questions that the words are slightly different, but it's not outrageously difficult. So in this situation, if I asked you which direction sodium diffused, the answer would be what? Diffused inside the cell. Which direction was sodium pumped? Outside the cell, right? Okay. And that sounds like memorization, but it's really not. After you get used to like playing with this a little bit, you're like, I understand both the words and the concept, and it's not as confusing initially, right? It just requires like a little bit of deeper level of understanding. So in the last class, since we got a couple minutes, they're a little bit behind you, so I'll stop now. In the last class, somebody came up and asked me, so how do you do well in physiology? So I don't know, it varies based on person, right? But FYI, can you start to see how the stuff that I talked about at the beginning of this lecture impacted the end of the lecture? Can you see that? Like things can't get across and then you have to try to figure out how they're going to move across. The entire semester is going to be like that. Like the stuff that I talk about today will be fundamental to you understanding the stuff that we do on Monday. So how do you get that level of knowledge and understanding? You either rewrite your notes critically, right, and figure out what you don't understand and come back and ask about it. Or I think study groups are really useful for physiology. If you are a smart person who thinks you know something well and you try to teach it to another smart person who's going to be supportive but critical, right? So the supportive part is really important, right? You don't say, that's stupid, jackass, and then you then you don't have a study group anymore. But you have to be kind of critical about whether your knowledge is deep. And if it is, you can move on. And then, of course, you have to be able to verbalize the whys. The whys are super important in physiology, right? Not what moves across the membrane, but why could this one and not that one, right? Okay. So when and if you are successful with study techniques, I'm going to ask you about them so that I can share them with the people who are having a little bit harder time, right? Because I would like for you to all be successful. Okay, I'll see you guys on Monday. Your quiz will be up by 7 tonight. 7. Because 5, it's almost 5. I always forget that it's really late when we're at